In this video, we're going to go over the basics of how you can design a drug. And drug design itself is a very expensive and long process. So each new drug, so you're looking at $300 million on average. Some anti-cancer drugs can actually go up to $1 billion to make. And the idea for making a new drug is first you start with an initial screen. So for example, you have an enzyme that you want to bind. So you're just going to try and bind a lot of different compounds and see what binds the best. And really what you're doing is you're measuring the inhibition constant. You're measuring Ki. Once you have found some drugs are sensitive, you can move on to an animal screen, more sensitive screens that look for toxicity, bioavailability, see how it works through your metabolism. But in general, through your assays, you eventually come up with what's called a lead compound. A lead compound is a candidate drug that has your desired effects, such as inhibiting an enzyme, and it binds very well. The KD for a lead compound should be less than 1 micromolar. And with that lead compound, you can refine it to make new drugs that hopefully bind even better. You can synthesize these drugs and you just repeat. So drug discovery is usually a cyclic process until you get eventually to clinical trials, which we'll talk about later in this video. So drug development today, one method that's used is called structure-based design or rational design. With this, you need to know the structure of your enzyme, and you want an in complex with a candidate to know how to develop a drug. And the reason for this is if you know how a candidate binds, you can better rationalize how to improve your drug so that it will have even better binding. Computational tools can really be helpful for this as well. So for example, let's say computationally, you get a lead and then you can do what's called molecular docking. So molecular docking, what you do is that you have your drug bind to your enzyme, and then you can change your drug in your computer and you can predict how better of a binding event do I get. By refining your structures in the computer, that will take less actual synthesis you have to do in the real world, which can cut down not only on time but also money. You can also do a target identification screen using a computer. That is, you can go and target parts of the drug that you wouldn't think normally would bind to anything. So a computer could find out new binding sites for a particular enzyme, and you can use these new binding sites to create new drugs. So not only can computers help with leads, it can also help with finding a new target for your drug. Another technique that is used in making drugs is called high throughput screening. Here, you synthesize your leads and you synthesize them in such a way that you can make a lot of them very fast. So, an example, we have some syntheses going on here where all you do is you change the R1 group, R2 group, or R3 group, and you can have a lot of different compounds. And then via a robot, you have a robot testing how well these drugs bind to your enzyme. And with this, you can do roughly 10,000 assays a day. You can test 10,000 different drugs onto your target per day, see which one's the best hit, and further refine from there. So with computational and high throughput screening, these are ways that we can narrow down what drugs are useful for us. When designing a drug, there are some things you have to be aware of, and that's its bioavailability. So it's not so much that a drug has to work, but it has to be able to find its target in the human body. And let's just consider oral drugs. Everything an oral drug has to overcome to find its target. 1, it has to be stable in the stomach. 2, it has to be not degraded in the stomach. 3, it has to be specific for your target and not bind anything else. 4, has to survive the liver, and the liver is where a lot of the detoxification happens for foreign elements, which a drug is. 5, it has to stay in your body long enough so it's not excreted rapidly by the kidneys. 6, it has to go to your target tissue from the capillaries. 7, if it's a brain, going to your brain, it has to pass the blood-brain barrier that blocks polar substances. And 8, if you're targeting the inside of a cell, it has to go through your membrane. So all these are challenges for drug design. So not so much that a drug works, but it also has to pass all of these. And pharmacokinetics is the study of how drugs interact with all these factors. And bioavailability is once the drug reaches target, how much of it actually survived the journey, or how much is actually there. That's bioavailability. Now let's say that you found a drug and you want to have this drug be used on humans. Well, before we can actually sell it, we have to go through clinical trials. We have to make sure these drugs are safe. And there are 3 stages in a clinical trial. Stage 1 is looking at the safety of the drug. Here you'll see what's the optimal dosage, how best to give the dosage, how often to give the dosage. This is usually carried out with 20 to 100 healthy individuals. But if the drug is already known to be toxic, usually the individuals will be volunteers who have this target disease. Stage 2 of clinical trials is the efficacy of the drug against the target disease into 100 to 500 volunteer patients. So now we're moving on people who actually have this disease. You're looking for side effects and further refinement of the dosage. And usually you do a single blind test. Generally, the patient doesn't know if they're getting the actual drug or a placebo. However, if the disease is life-threatening, it's unethical to give a person a placebo. So instead of a placebo, you'll use the best available treatment to see if your drug works better. Phase 3 of clinical trials is looking for any adverse reactions from long-term use and to make sure this drug actually works. So you're bumping up to 1,000 to 5,000 people. This is a double-blind study where the person given the drug and the person receiving the drug do not know if it's a real drug or it's a placebo. This is to eliminate all bias. The results we see cannot be biased by any beforehand knowledge. And just to give you a scope of this, of 5,000 drugs that are made, only 5 will go to clinical trials. And the 5 that go to clinical trials, only 1 will usually make it through all 3 stages. And this process from initial screen to development to actually being available for people to use takes anywhere from 10 to 13 years. And like I said, $300 million to some drugs costing $1 billion. So it's a very time-intensive and money-intensive process to get pharmaceuticals. And not only does it take a long time, sometimes drugs can have an adverse reaction that we don't see in the clinical trials. And there are many different ways why an individual can have an adverse reaction. This can be genetic differences in your disease state, if you're taking other drugs, age, sex, or environment. And the molecule of protein that's usually responsible for adverse reactions to drugs is cytochrome P450. P450 is found in your liver. It's found in other places too, but is about 2/3 of the 450 is found in your liver, and it's actually found in all types of organisms. Humans have 57 different types of P450, and P450 usually helps to detoxify foreign substances. But let's say you're taking 2 drugs at once, and these drugs weren't tested in the clinical trial. And let's say drug A inhibits P450 so it can no longer work. Well, let's say you're taking drug A with drug B. Well, drug B is normally destroyed by P450. But if you take drug B alone, enough of it still gets to your proteins that is effective. Well, if you take drug A and drug B at the same time, then what you're doing is you're increasing the bioavailability of B. That is, more B is being delivered than we saw in the clinical trials. And this can lead to any number of adverse reactions. So that's just a further complication of drug development with P450. And that's it with this video and all the videos in this class. So thank you very much for listening. Congratulations for making it this far. And I will see you in class.